So I am going to move on and invite our next speaker. The presentation is titled "The Use of Novel Technologies to Better Understand Perching Biomechanics and Keelbone Damage in Enriched Housed Laying Hens." Emily Depaoli, University of Alberta. Please go ahead. Depaoli. Apologies.
Okay.
Thank you.
Hopefully you can see that okay. I'm going to assume that's a yes.
Good.
Yes.
Sorry, it looks good,
Okay.
Emily.
It's okay. Thank you. So, hi, my name is Emily Depaoli, and I'm a current master's student at the University of Alberta in the Department of Agriculture, Food, and Nutritional Science, and I am a member of the Ethology Research Lab under the supervision of Dr. Clover Bench. Today, I'll be talking to you about my research, the use of novel technologies to better understand perching biomechanics and keel bone damage in enriched house laying hens. So the keel itself is a part of the sternum, and the sternum is a modified breastbone that is present in all modern birds, but the keel is only present in birds that fly because it is an attachment site for flight muscles. Keel bone damage can be classified into two categories: deviations and fractures. Fractures, as seen in the first image, are fragmented sections, while deviations, seen in the second image, are abnormal shapes not associated with a fracture, and they are often seen as a C or S shape in more severe cases. There are many potential negative impacts of keel bone damage, including reduced egg production, lower quality eggs, parameters such as shell sickness and shell breaking strength. We also see altered locomotion. They move more slowly around their enclosure, and they rest more while either standing or sitting. They also experience pain from keel bone damage. Overall, these parameters indicate reduced welfare. Oops, sorry. So conventional cages have been shown to limit the expression of natural behaviors and restrict movement. The industry is currently shifting to enriched housing to improve the overall welfare of laying hens. However, there has also been a notable increase in keel bone damage in enriched housing due to the increased opportunity for movement. Understanding how to mitigate the increase in keel bone damage is currently vitally important for the industry. Perching itself is an important natural behavior that has been noted in the living ancestor of the modern laying hen, the red jungle fowl. However, the red jungle fowl has a greatly reduced level of keel bone damage compared to their modern counterparts, which is a potential indicator that artificial selection has had a negative impact on modern layards and their keel bone health. So the current objectives of our research are to discover which perching phenotypes are associated with better foot and keel health, as well as stronger bones. We want to use 3D kinematics to assess the biomechanics of perching behavior in pullets and laying hens. We want to determine what genomic sequences contribute to specific perching phenotypes, and we want to understand how different factors such as weight, strain, perch shape, and rearing environment impact perching and keel bone damage. So what is 3D kinematics? 3D kinematics utilizes near-infrared stroboscopic cameras that pick up reflections from markers that are placed on important skeletal landmarks. These recordings will provide us information about balance, movement, joint angles, and stabilities while birds perch. Infrared thermography utilizes cameras that detect heat from an object and turn that into a temperature distribution image. By assessing footpad pressure points and how the posture of the laying hen is impacted by perching, we can better understand how pressure is exerted on the keel and foot pads. DEXA stands for dual X-ray absorptiometry. It is typically used in human medicine to diagnose osteoporosis. It can provide us information about important health markers such as bone mineral content. So in the first trial of my study, we will be utilizing a variety of strains of laying hens to understand how genetics influence perching. By raising the laying hens in enclosures that do not have perches, we will create a baseline for what innate perching behaviors are present. and they will not have the opportunity to learn how to perch in their enclosures. Recordings will be taking place at 18, 29, and 70 weeks using round and mushroom perches, while DEXA scans will be conducted post-mortem. We have selected these time points for data collection due to the classic egg production curve, as we have points at the beginning of lay, the peak of lay, and the end of lay. Each bird will be phenotyped based on the data we collect surrounding their perching biomechanics, footpad, and keel bone health. We can then perform a genome-wide association study based on pre-selected candidate genes to help identify any important genetic sequences for improved perching ability, keel bone, and footpad health. In trial 2, we will determine how different environmental factors impact perching behaviors and keel bone damage. Based on the information we collected in trial 1, we will select the 2 best strains of commercial layers, one white and one brown, that have demonstrated an ideal perching phenotype. By utilizing a variety of metrics, we will determine how strain, weight, pullet rearing environment, and perch shape impact keel bone damage. So our research will impact codes of practice. We hope to help fill gaps that were identified in the unfact pullet and laying hens code of practice. We want to help producers effectively transition from conventional to enriched cages by providing information on management practices to reduce keel bone damage. We want to help researchers by highlighting the use of novel technologies within fields such as animal agriculture, and we want to help identify genetic sequences for improved health and welfare of laying hens for genomic companies. I'd like to thank our funders, the Alberta government, Egg Farmers of Canada, and Egg Farmers of Alberta, as well as everyone that has helped make this project possible. If you have any questions, now is the time. You can also email me. Thank you.
Thank you, Emily. Well explained. Reminder that if you have any questions, please type them into the Q&A box now and we'll read them out. I was interested in knowing, while people are thinking up their questions, you talked about the novel techniques, and I'm really enthralled by that, because to me it takes out the subjectivity, right, when you're using equipment to do the measurements. Do you see any opportunities for, at the producer level, at the farmer level, using versions of those kinds of techniques on the farm to be able to potentially have early indicators of issues and being able to take action earlier potentially?
My lab actually does a lot of research involving infrared thermography, more on a producer level, to utilize that technology to identify issues such as like disease and stuff like that, kind of at earlier stages. So that is research that my lab does.
Excellent, great. Okay, I am waiting to see if there are any questions. I am not seeing any at this point. We do have a couple of extra minutes, and after that, we do go for a break before we have our next session. All right, well, I'm not seeing any at this time. I think we will leave it there for now, and I want to thank you so much, Emily, for your presentation.